Okay, thank you everyone. Good afternoon. I realize I'm standing between you and the break, but I am delighted to be here. I'm Kristen Urema, currently serving as president on the cell therapy side for Poseida, and I'm very happy to have the opportunity this afternoon to introduce you to our company. So, some forward-looking statements that we're all acquainted with. So, Poseida is a group team of very passionate and dedicated folks. We are located just a few minutes south in La Jolla, California. And our mission is really to advance a new class of cell and gene therapies. So we are very proud, I would say, not ashamed at all, very proud to call ourselves a platform technology company. That is because we believe that the suite of technologies that we have developed internally are very powerful, very unique to Poseida, and really set us up well to be in a position to discover and develop new therapies, both in allogeneic cell therapy as well as in vivo gene therapy. So you see there, we are active in two major parts of what we are all here at the meeting on the Mesa and part of ARM to try to advance. On the cell therapy side, we are firmly in the allogeneic camp. The company did start with some autologous programs, but we have shifted that focus entirely to allogeneic cell therapy. We have a major partnership with Roche for hematological malignancies. And earlier this summer, we also were very pleased to announce a strategic investment by Astellas. And then in the gene therapy side, we are really focused on trying to move beyond viral vector to offer new types of gene therapy in the future. So let me start by telling you a little bit more about our platform technologies, and then we will go on to talk about the pipeline and the programs that these have enabled. So starting with gene insertion. So our gene insertion technology we call Piggyback, and it is transposon-based. So it is intrinsically non-viral. It's highly efficient and also can carry a very large cargo capacity, much larger than what we can do with viral vectors, for example. We are focused on T cells today, and the pipeline CAR T that I'm going to talk to you about are all obviously T cell therapies. But Piggyback is actually an approach that can be used in a wide range of different cell types, and we have done work investigating that applicability in our labs. We often pair that technology with our gene editing technology that we call Cas-CLOVER. Cas-CLOVER is very precise. It has the ability to edit resting cells, and I'll give you more of a schematic and talk to you a little bit more about that one in just a second. We're also exploring gene delivery technologies. We have an in-house gene delivery discovery group. We work in LNPs and lipid discovery. And our focus is really to enable long-term stable gene expression. And ultimately, while we work today in hybrid approaches using a combination of viral and non-viral, we would really like to get to a fully non-viral approach where we can do gene therapy or deliver genes both ex vivo and in vivo. At Poseida, we love to innovate. We are always innovating and never rest. So there are many technologies that we have in development. I'm not going to talk a whole lot about those today, except for the one that's at the very bottom of the page, which we call our booster molecule. And that is a technology that we have developed that really helps us manufacture, particularly allogeneic T cells at a high yield. And if you're a cell therapy developer yourself, you know that trying to manufacture T cells at scale after you've taken out the endogenous TCR can often be quite challenging, and we believe we have solved that problem with our booster technology. Okay, so a little bit more about Piggyback. So you see the cartoon there. So Piggyback is a transposon-based non-viral technology. And it works with a plasmid. You see the plasmid there carrying the DNA cargo, flanked by inverted terminal repeats, and the super piggyback transposase that I really wish looked like Pac-Man in real life, but it's good for cartoon purposes. And we think of this as a cut and paste technology. So super piggyback will clip out the DNA cargo of interest and then integrate it stably into the host genomic DNA. So advantages of this are, as I said, it works in a wide variety of cell types in addition to being intrinsically non-viral. And we believe that that's going to demonstrate multiple safety as well as cost benefits. How is it particularly relevant for cell therapy? Well, as I'll tell you in just a moment, we focus in our CAR T work on a particular type of T cell that is the T stem cell memory cell. And one of the great advantages of piggyback is that it will preferentially transpose naive T cells. So it works well in resting T cells, and so we can, through our system together with Cas-CLOVER, really preserve the T stem cell memory phenotype. And then also, the very large cargo capacity allows us to engineer in a huge amount of functionality into our T cells. On the gene therapy side, it's, as I said before, it's a stable integration with long-term expression. So this is very useful if, let's say, you want to develop a therapy for kids and target the liver. Well, you can do that. You can target a dividing tissue like juvenile liver with this technology. And again, the large cargo capacity just enables us to deliver very large genes. So Cas-CLOVER is our gene editing system. And this uses 2 deactivated Cas9 enzymes fused to the CLO-051 enzyme, and we call that fusion molecule Cas-CLOVER. You see there that it requires 2 guide RNAs, one on either side, and it will not make a cut unless both of those guide RNAs are in place. So what we see is low to no off-target cutting. It's a very high-fidelity system, easy to use, highly efficient, and we see greater knock-in efficiency than with Cas9, for example. So that's the toolbox in a nutshell. We are not just a platform technology company, although we're proud of that, but we have a rich pipeline, and we're excited about the programs that we're advancing in the clinic. So this is a look at our allogeneic CAR T pipeline, where we'll start first. You can see we're active in both solid tumor and heme malignancies. We retain all the rights to our solid tumor programs, and then in heme, our 2 lead programs are licensed to Roche, and Roche has an option on a couple of the others. So let's talk about those a little bit more. So we're focused on T stem cell memory cells. Why? Well, we think that these cells are likely to have both safety and potentially efficacy advantages as well. They are self-renewing. They engraft. Think of them as a prodrug. So you engraft the less differentiated T stem cell memory cell, and then it will self-renew, divide, have a long life, differentiate, and then act as a CAR T. Cas-CLOVER allows us to be fully allogeneic by editing out the TCR as well as addressing MHC1, so that tackles both sides of the alloreactivity concerns. And then our booster molecule that is added at the same step that we do all of our other editing work helps us reach the kind of manufacturing yields that we need. And we have been recently discussing that we're seeing triple-digit dose achievements from a single batch, a single starter leukopak of cells. The T stem cell memory cells, I won't cover all of this, but if you're familiar with T cells, you know there's a spectrum of differentiation. Our product is essentially T stem cell memory cells and some of the central memory cell type. That has been correlated in the clinic, not just through our work, but with a growing body of literature in the field. And we think that this, starting with the prodrug approach, gives the cells a little bit more time. You're not hitting the tumor with a very hot, very active effector cell type CAR T, and so that should lead to less CRS and other sorts of adverse events. Here's a blow-up of the cargo from the piggyback transposon, and let me just touch on this for a second, because this shows you some of the functionality that we put into our products. So all of our products include a proprietary safety switch, a very rapid-acting, kinetically favorable safety switch. We hope that we don't need to use it, but if we do, it's there. We can also carry multiple CARs, full-length CARs, and we've shown 2 just for illustration, but we could do more. And also a selection gene that we use in purification. And if you can see that little bullet at the bottom there, you see that our product is essentially 100% T cells expressing CARs. So we touched on Cas-CLOVER a little bit already. Let me talk to you about the first program, which is a solid tumor program against MUC1C. So mucin-1 is not a new target for endothelial cell tumors, but our program is able to target the C-terminal region. This program is in phase 1 dose escalation. We reported data last year, early data, in 6 evaluable patients where we saw a very favorable safety profile and also a partial response in a breast cancer patient. And breast cancer is one of the tumor types where we know MUC1 and MUC1C is an expressed target. So we'll be looking forward to our next data update on this program. Our other program in phase 1 dose escalation actively currently is P-BCMA Allo-1 against B-cell maturation antigen. This program is for relapsed refractory multiple myeloma, at least to start with, and patients have to have received at least 3 lines of therapy, including a proteasome inhibitor, an IMiD, and an anti-CD38 antibody. So this is, again, a safety and dose escalation study ongoing. Last year, about a year ago, at ESMO IO, we presented our first data in 10 evaluable patients. We found no dose-limiting toxicities, CRS or graft versus host disease or ICANS, nothing like that, and some early signs of efficacy. We are looking forward to providing a data update on this program at a medical meeting later this year, likely ASH, so stay tuned for that. Then we're also looking forward to our third program. This is the second program in the Roche collaboration, and this is our first dual CAR T. So I just want to be clear, this is not a mix of 2 different single CARs in a product or anything like that. Because of the cargo capacity of piggyback, we're able to put 2 full-length CARs into the single T cell. So we believe this is the first allogeneic dual CD19, CD20 targeted CAR T. We think that's important because a common escape mechanism in B-cell malignancies for relapse is loss of the CD19 antigen. And there are auto programs ahead in the clinic that have showed, pretty exciting data. So we're really looking forward to seeing what this allogeneic program can do. So in a nutshell, how are we different? What do we see as our advantages in developing allogeneic TCR-T and CAR T? Well, it really starts with those T stem cell memory cells and our ability to produce a CAR T product that is, highly, highly prevalent in that very meaningful cell type. We are fully allogeneic, and our experience has shown the safety we've been expecting with that to date. We've talked a lot about the cargo capacity with piggyback, and we also are looking forward to incorporating other technologies, such as armoring technologies. Let me touch very briefly also on gene therapy. So the gene therapy programs are earlier. They are in a preclinical stage. We put out quite a bit of data at this year's ASGCT. You can find that available on our website. And our goal is really, as I said, to move ultimately to a fully non-viral approach that can bring a new look at gene therapy to patients in rare disease. I'd also like to mention that the gene therapy programs are available for partnerings, potentially. So if anyone has interest in that, let me know, and I'm happy to put you in touch with our BD team. So as we think about where we are, you can see we've been pretty happy to achieve a couple of our milestones this year, looking forward to providing further data updates. And again, if anyone has interest in a conversation about gene therapy, let me know. So thanks very much for your attention. Let's go get some coffee and a break. And it's great to be here with all of you at Meeting on the Mesa. Thanks.